Hi, Ron. My name is Kimberly Skead, and I'm a PhD student at the Ontario Institute of Cancer Research. I'm really excited to have the opportunity to chat to you today about some of my PhD work, which is looking at the impact of selection on shaping our blood health as we age. My work is particularly focused on the application of a population genetics lens to understand blood evolution at an individual level. The reason why we can do this is because the entire human hematopoietic hierarchy is rooted in a small population of stem cells which reside in our bone marrow. These cells are highly productive and are ultimately responsible for all the mature cell types in our blood. Naturally, because they are so highly productive, they need to be very tightly regulated in order to maintain the composition of the mature blood pool. Something which people have been paying a lot of attention to lately, though, is this notion that as we age, we start to accumulate somatic mutations either in our stem cells or in early progenitor cells, which confer a growth advantage to those cells, causing them and all of their derivatives to become disproportionately represented in the mature blood pool. Typically, when we see that one clone has become represented at a frequency of greater than 2%, we say that individual has clonal hematopoiesis. And because this is a phenomenon which has been closely linked with age, it has been termed age-related clonal hematopoiesis, or ARCH. This is something which is very common in the adult population, but interestingly has been found to associate with around a 10-fold increase in likelihood of progressing to cancers or cardiovascular disease. We don't really know why some people progress to disease and why others remain healthy, but one of the working hypotheses is that in those individuals progressing to disease, they're acquiring a driver mutation in a known cancer gene, for example. But if we look at the proportion of individuals in the population who are healthy and harbor a mutation in a known driver gene, shown here in gray, compared to the proportion of individuals who eventually progress to disease, shown in red, those numbers look quite different. And obviously, there's some fairly major implications to this. If we start responding to that gray group, we're going to end up hugely overtreating a large chunk of the population that never really required that therapeutic intervention in the first place. But if we don't respond to this red group, we're going to miss out on an incredibly valuable chance to capture disease in its infancy when treatment outcomes are more likely to be effective. What we think is that the background upon which these mutations arise matters. By that, I mean, we have a very good idea of what positive selection looks like when it's acting to increase the frequency of a driver or advantageous mutation in a population. We also have a very good idea of what negative selection looks like when it's removing mildly damaging mutations in order to maintain the integrity of our tissues. What we don't really understand is what happens when we see a mutation under positive selection arising on a background where there are already a number of mutations under negative selection. We might start to see competing evolutionary dynamics, which could be functioning to inhibit those disease-associated clonal expansions. Of course, we also need to consider a scenario wherein any changes in variant allele frequencies are occurring purely by chance or through genetic drift. I'll mention here that cancer evolution is most frequently studied through this lens of positive or neutral evolution only, and we often ignore the impact of negative selection on shaping cancer outcomes. Our team has previously shown that all of these competing selective pressures exist in blood in a paper which came out in Nature Communications in 2021. Here, we built a machine learning classifier to evaluate the distribution of somatic mutations in a blood sample, and from that predict those different selective pressures in blood. We applied our machine learning classifier to a cohort of blood sampled from healthy individuals, as well as blood sampled from individuals about 7 years prior to their progressing to acute myeloid leukemia. What you can see here is that the vast majority of individuals are rejecting those neutral and negative only models of blood evolution, and we are seeing signatures of negative selection or combination models of evolution being the most predominant mode of blood evolution. And interestingly, these different selective pressures do appear to be playing a role in preventing progression to acute myeloid leukemia. You can see here that these individuals who fit those negative or neutral models of evolution are in fact remaining AML-free for significantly longer than the individuals fitting positive or combination models of evolution. Right around the time that we were working on this question of how does our blood evolve, a fascinating paper was published out of Alkis Price's group, which reported the presence of large structural changes in the blood of a population cohort in the UK. Through looking at genotyping array data and contrasting the intensity ratios and B allele frequencies from that array data, their team was able to call somatic structural changes in the blood, which have been termed mosaic chromosomal alterations, or MCAs. Typically, these MCA events tend to fall into 3 categories: instances where we are gaining or losing chunks of our DNA, or instances where we are losing heterozygosity at certain regions termed CNLOHs, or copy number loss of heterozygosity. But the key finding, which really jumped out at us, was this one, that 5% of the healthy population harbor one of these massive structural changes, which can really get as large as an entire chromosome in size in their blood. From a selection standpoint, this really doesn't make too much sense because presumably such large-scale changes would be quite destructive and would be removed from the mature blood pool by negative selection. So we wanted to dig a little bit deeper into this question, so we turned to the Canadian Partnership for Tomorrow's Health platform, or CanPath, which is a large population cohort hosted here at the University of Toronto, which has collected self-reported health outcomes and biological samples, thus enabling genomics profiling for around 330,000 Canadians. Through CanPath, we were able to access genotyping array data for about 14,000 individuals to call somatic MCAs in the Canadian population. Here, I'm showing you a circus plot where you can see the chromosomes indicated along the outer ring and the locations of loss, gain, and CNLOH events indicated by the bands of blue, green, and orange. One of the benefits of CanPath is that we have access to a variety of different genotyping array technologies, which lets us start to tease apart some of the impacts of those technologies on our ability to make these biological inferences. For example, here, you can see that right away, we are capturing a much, much higher prevalence of somatic MCAs in the general population, with up to 14% of individuals harboring one of these large structural changes in their blood. We think that this difference is largely due to the fact that we are using a denser genotyping array and have more resolution to capture smaller MCAs. And as you can see here, where I'm showing you the density of each array along the bottom and the distribution of MCA lengths along the y-axis, we do find that we're able to identify those smaller MCAs that would otherwise have been missed with our denser genotyping arrays. That said, the length of an MCA actually becomes quite interesting from a selection perspective. Presumably, large MCAs are going to implicate more genic material and be more damaging, or rather have a larger impact on the fitness of a cell. We can test this association quite directly through comparing the length of an MCA with the cell fraction, or rather the number of cells in that individual's mature blood pool, which are estimated to harbor that MCA. In the absence of selection, where these two variables are unrelated, we might expect to see something like this, where there's no real pattern between those variables, length is having no impact on the expansion of clones harboring those MCAs. However, we are instead capturing this really nice relationship where we find that the size of those MCAs are negatively correlated with their frequency in our blood. We think that negative selection is playing a role in limiting the expansion of clones harboring these large variants, and it's only those much smaller MCAs which are able to rise to a higher frequency in our blood. Of course, the size of an MCA is not the only consideration with respect to the impact of the MCAs on cellular fitness, and we need to think about which areas of the genome these MCAs are impacting. To address this, we implemented a genome-wide hotspotting approach where we quite simply scan the genome looking for regions where we capture a disproportionately high burden of MCAs. Here, you can see each chromosome indicated as a box with a genomic location along the x-axis and regions which are significantly enriched for structural changes highlighted along the y-axis. For example, we can zoom in on chromosome 13 here, and you can see that we're capturing an enrichment of loss events at this location. And interestingly, this enrichment peak is actually popping up over DLU2, which is a region which has been implicated in ARCH or precancer phenotypes previously and is known to play a role in the development of chronic lymphocytic leukemia. And we actually find that these MCA peaks are often accumulating over genes which are either ARCH or cancer associated. For example, here you can see this chromosome 2 peak, which is hitting genes like ASXL2 and DNMT3A. Of course, this preponderance of MCA peaks at genes which are implicated in cancer raises an interesting question about whether or not these individuals are in fact at an increased risk of progressing to cancer, or blood cancer in this case. Again, this is something that we can test quite directly in CanPath, as all of our participants have consented to administrative health linkages, meaning that we're able to capture clinical outcomes at the ICD code level. To give you a sense of what this looks like, we have participants who are healthy at the time of enrollment and who have remained healthy throughout their participation in the cohort. We can also identify individuals who joined the cohort with a history of cancer. However, we have this third group here, which are these individuals who are completely healthy before joining CanPath. They came into the cohort, they gave us a blood sample, and years later were diagnosed with cancer. From a research perspective, this group is immensely valuable because it gives us this wonderful snapshot into whatever is going on in that individual's blood system, sometimes years before that diagnosis. To identify these three groups in the CanPath code, we link the Ontario arm of CanPath to Cancer Care Ontario to capture these cancer outcomes. And when we look at patterns of MCA accumulation across these groups, we find that individuals who harbor an MCA are in fact at a significantly greater risk of progressing to cancer, and that this risk really scales with the number of MCAs per individual. You can see here how this hazard ratio is really increasing in individuals who have 1 to 2 MCAs and individuals who have over 3 MCAs in their blood. As we're capturing this link between these somatic events and disease risk, we wanted to interrogate the functional impact of these structural events on intermediate phenotypes such as gene expression. We can do this in CanPath as we have overlapping array data and RNA sequencing for just under 1,000 individuals. This lets us start to ask some questions like, what is the impact of somatic mosaicism on gene expression variability broadly? And second, how might these somatic events, which we've only been able to capture very recently, be confounding our understanding of the germline contribution to gene expression variability? To tackle this first question, we performed a burden analysis to determine the impact of somatic mosaicism on the transcriptome. Quite simply, we rank ordered individuals by expression levels for each transcript and captured the distribution of somatic MCAs across gene expression percentiles for all genes impacted by an MCA in the cohort. What we find is that gene expression is not able to recover from loss events, and we see a greater burden of somatic MCAs at the extremes of gene expression. Conversely, we find that when individuals gain additional copies of DNA, we don't see an increase in gene expression, but rather we see that the greatest burden of gains occurs around that 50th percentile mark, or rather the population average level of gene expression. We think that this might indicate the presence of stabilizing selection, where even with an additional copy of DNA, average levels of gene expression tend to confer a higher fitness advantage. Finally, we wanted to capture the impact of MCAs on confounding measurements of germline factors contributing to gene expression variability. There are a couple of canonical approaches that we use to quantify this, such as allele-specific expression and EQTL mapping. To capture the impact of somatic MCAs on driving gene expression variability, we developed a model to assess the percentage of sites in a given genomic region exhibiting allele-specific expression and looked at how this varied in regions impacted by somatic MCAs. As you can see here, compared to that dashed line, which indicates the percentage of ASE in regions not impacted by MCAs, we are observing inflated estimates of ASE in regions impacted by loss in CNLOH events by about 7% and 3% respectively, due to the previously unaccounted for loss of a parental allele. And we see a similar thing with our EQTLs. Here, we developed a pipeline to perform somatic copy number adjusted EQTL mapping. When we compare the effect sizes of our MCA agnostic and copy number adjusted EQTL calls, we find that MCAs impact our ability to correctly infer the effect sizes of up to 1 in 3 EQTLs. And so broadly, what this is suggesting is that there is a component of what was previously thought to be inherited variation in gene expression that can actually be explained by these somatic events. I'll end off by highlighting some of our key takeaways. First, we're finding that MCA-associated ARCH is much more common in the population than previously estimated. These events do appear to be preferentially retained in regions implicated in ARCH and cancer development and do appear to impact the risk of cancer progression. And we don't think that this association is random, and we are capturing signatures of selection, which we think are playing a role in maintaining somatic variability both at the DNA and RNA level. And finally, what I really think that this work is highlighting is the importance of applying more of a multi-omics lens through which to understand the impact of somatic variability on cancer risk in the population. With that, I'd like to end off by thanking many people, most importantly, the participants of the CanPath and EPIC cohorts who really enabled this research, as well as all of our funders. Finally, a big thank you to my advisor, Philip Audella, as well as many of our lab members and collaborators. Thank you.